Okay, now on to part 4. Let's talk a bit more about this idea of a genetic code and translation. Now recall that amino acids are coded in triplets, or sequences of 3 nucleotides, like you see here in this diagram. Notice the difference in the base coding from DNA to mRNA. Remember that uracil replaces thiamine. For example, you see the triplet coding for the amino acid trypsin. So trypsin is TRP right here. Here's the amino acid trypsin. And you see that its code is UGG. Well, the DNA version of it is ACC. Whoops. So this is the DNA version of it. And I know it's the DNA version of it because there's thymine right there. And then mRNA converts now the DNA coding, or codon in this case for trypsin, into UGG, which is now the mRNA version of the trypsin code. Now phenylalanine, on the other hand, here's phenylalanine right here. All right, so there's the 1, 2, 3 triplet code for trypsin, and now we're on to the 1, 2, 3 triplet code for phenylalanine. Phenylalanine has a DNA code of AAA. So if we look up here, here's the 3 A's for phenylalanine. And the mRNA coding is UUU. Now write in your notes that genetic information is encoded as a sequence of non-overlapping base triplets. Non-overlapping base triplets. What do we mean by non-overlapping? It means that this now doesn't overlap now with the codon for trypsin. So clearly delineated 3-letter codes for each amino acid. And that the gene that determines the sequence of bases along the length of an mRNA molecule carries this code. So look up here now at this diagram. Here's gene 1 now. See the little square? Now we've got that gene 1 blown up here. And now you see the codes for these amino acids. So this gene 1 would code for some kind of protein. And then these would be the amino acids that this protein now would be built on. And over here you see transcription now, the building of the mRNA molecule. And now we're on to translation, where we actually bring the protein parts and the instructions together to build that protein. We call that translation. So get that in your notes. So, write in your notes that codons are a 3-base code for the production of a specific amino acid, which is a sequence of 3 of the 4 different nucleotides. And since there are 4 bases and 3 positions in each codon, there are 4 times 4 times 4, or 64 possible codons. Make sure you get that in your notes. Also get down that that's 64 different codons, but only 20 different amino acids. Therefore, most have more than 1 codon. That makes sense. Now, in addition, write that 3 of the 64 codons are used as stop signals, and you see them right here. So 3 of these 64 are used as stop signals. And one of them is used as this start, and we're going to see that here in a second in translation. Now, the stop signals are found at the very end of each gene and mark the end of the protein. And again, you see them here in the diagram. So a codon in messenger RNA is either translated into an amino acid or serves as a translational start-stop signal. Also write that in your notes. Now, one codon is used as a start signal, and it's at the start of every protein. And that start signal is universal in all living organisms. So finish off this slide with that in your notes. So the start codes are universal in all organisms. Now let's talk about the next important form of RNA, which is transfer or tRNA. So write that in your notes, transfer or tRNA. Now first let's look at the structure. Write in your notes that the 4 base paired regions and 3 loops you see here are characteristic of all tRNAs, as is the base sequence of the amino acid attachment site at the 3' end. So let's look at this structure. I'd like you to get it in your notes. So I want to explain it first and then have you write it in your notes, because it's important for you to understand that transfer on RNA looks completely different than messenger RNA. So here's your amino acid attachment site. And it's just exactly what it's going to do, is attach to an amino acid. So great name. Now, down here you see the 3 loops. That's how you know this is a transfer RNA molecule. So on an exam, they may put some diagrams of different RNA or different molecules, period, and you have to identify which one is transfer RNA. These 3 loops are classic transfer RNA. Now, and here's your anticodon down here, AAG, and we're going to talk about that here in a second. So get this diagram in your notes and label it correctly. Now, the anticodon triplet is unique to each RNA type. Write that down. So the anticodon triplet, so in this case it's AAG, is unique to each tRNA type. Anticodon triplet. Big name, huh? Well, if mRNA is carrying the triplets for each amino acid, then we couldn't have tRNA have the same triplet for every codon, right? Now, we call it an anticodon because it uses the RNA base language to translate each amino acid. Write that in your notes. For example, in the previous slide, the DNA code for trypsin was ACC, which meant that the mRNA code for that amino acid was UGG. So, it makes sense that the corresponding tRNA anticodon, where you see here anticodon, would then be ACC again, because now we're translating in RNA language. The asterisks you see here mark bases that have been chemically modified to a characteristic of tRNA. Do you see these little asterisks right here? Those are areas that have been chemically modified. Also write that tRNA consists of a single RNA strand that is only about 80 nucleotides long, and that each tRNA carries a specific amino acid on the 3' end and has an anticodon on the 5' end, like is shown here in this diagram. In addition, write that the anticodon, down here, anticodon is the 3 RNA bases that match the 3 bases of the codon on the mRNA molecule. Now, it's not going to be AAG on the mRNA molecule. It'll be RNA code, but not AAG, and you can figure out what it would be on the mRNA code. Now, then finally also write that a special group of enzymes pairs up the proper tRNA molecules with their corresponding amino acids. and that tRNA brings the amino acids to the ribosome. Now, it should be no surprise that it's enzymes that do this. Now, remember, enzymes are proteins. So it's interesting that we have proteins that are already formed. Here we are again, working on the formation of proteins. That's how important proteins are in your body. So the enzymes now in this situation pair up the proper tRNA molecules with their corresponding amino acids, and then so that TNR can bring those amino acids to the ribosome. So remember now, in transcription, we got mRNA, we actually made an mRNA that copied DNA, the instructions, right? Now we've got a tRNA molecule that's going to bring those amino acids together with the instructions at the ribosome to build a protein. So that's where we're going. Now, in that last slide, we looked at the tRNA structure in 2 dimensions. If we look at it in 3 dimensions, we see that the tRNA molecule is roughly L-shaped. So here's our L shape right here. It's roughly L-shaped. You see an artist's rendition of this molecule here on the right. So remember, our list of 4 things involved in transcription. Let's review them again. First, there was mRNA, which then gives rise to transfer RNA molecule. Next, let's go to the place where protein synthesis takes place. Where is that? Yes, the ribosome. Okay, remember that ribosomes are one of the key organelles that the cell possesses. Remember, an organelle gets its name because it's one of the cell's organs. You probably should write that down. Now, what actually happens at the ribosome? First, write that ribosomes facilitate the specific coupling of tRNA anticodons with mRNA codons during protein synthesis, in that the 2 ribosomal subunits are constructed of proteins and RNA molecules named ribosomal RNA or rRNA. Now that's a lot, okay? So now we're introducing another RNA molecule, ribosomal or rRNA. So the 2 ribosomal subunits, mRNA and tRNA, are constructed of proteins and RNA molecules named ribosomal RNA or rRNA. So here's your mRNA molecule. Here's your tRNA molecule. So when those get coupled together now, this becomes now a functioning rRNA molecule or ribosomal RNA molecule. Now this particular diagram is a model of a bacterial ribosome, and it shows its overall shape. So remember, eukaryotic, prokaryotic. Bacteria is prokaryotic, and then our cells are eukaryotic. Now the eukaryotic ribosome is very similar. So remember that a ribosomal subunit is an aggregate of ribosomal RNA molecules and proteins. Okay, I'd like you to get this diagram in your notes. So go ahead and get this diagram and label it correctly, and then let's talk about it. Okay, so a ribosome has an mRNA binding site. You see that right here, an mRNA binding site. First of all, you should notice that it has 2 separate parts, a big part right here and a small part right here. So they call this the large subunit, and they call this the small subunit. Pretty descriptive in my opinion. So it has an mRNA binding site and 3 tRNA binding sites known as the A, P, and E sites. A, P, and E. You see those here. This schematic ribosome will appear in later diagrams, So again, make sure you label all of it correctly in your notes when you have that diagram in there. Now, let's go over each part of the ribosome. You can see that each ribosome has the two main areas, so make sure you have those labeled. Now, the P site. So let's talk about the P site first. And you have this peptidyl tRNA binding site. So you need to write that down and make sure that's labeled correctly. You have the A site, the amino acyl tRNA binding site. And then you have the E or exit site. So make sure you have those labeled correctly. Now you also see the mRNA binding site. Okay, make sure you have all of this labeled correctly. We're going to refer back to it in our discussion of ribosomes. Okay, we're almost there. We're to the point now where we have the instructions with us, and now we have the ability to have the amino acids with us at the ribosome. So once we have those two things, now we go through the actual building of the protein, which is our next step. So take a picture of your notes, submit them to Moodle, grab that study guide, and then answer the questions, and then I'll see you next time on Part 7. Bye.